Hello, my name's Zoe Barker. I'm one of the researchers that has been working on the Diverse Forages Project, and I'd like to take you through some of the results from the project. So these results in this presentation are from the agronomy plots that we've had across the different locations around the south of the UK that Chris has introduced you to already. So Chris has introduced you to the locations that we used and the seed mixers. So just to remind you, at each of these locations we took a number of different measures, including assessing the botanical composition of the plots and also measuring the biomass as well. So these results here are from the main 4 study locations, so the 2 plots in Berkshire and the plots in Cornwall and Devon. And first of all, you can see that the weather conditions in years 2 and 3, where we had 2 significant drought periods, affected the overall yields, and they were lower than that of year 1. And also in year 1 we had better yields for perennial ryegrass than we did for the multi-species swards. However, this trend is reversed by the time we get to year 3, and this could be partly associated with weather conditions affecting the ryegrass negatively, but also the fact that we've now had 3 years for the effects of the nitrogen fixing legumes in the mix to take hold. So as well as the different locations, we also had plots that were planted within the crops research unit of the University of Reading, but on very different soil types. So we had a very dry, gravelly site, which we're calling our dry plot, and we also had a very wet, low-lying area by the side of a pond, which we call our wet plot, and also one in between. So from this, what we can see is the first year results are quite similar to that of the biomass that we've just looked at. So perennial ryegrass was doing slightly better, but yield was down compared to the first year, and is even more affected when we look at years 2 and 3. And when we've got years 2 and 3, the effect of treatment is not significant. So there's no difference between the perennial ryegrass or any of the multi-species swards. If we look at all of the years but look at the different sites, so the dry, medium and wet, you can see that the dry site is yields lower than the medium and the wet sites, and that first year pattern of perennial ryegrass doing slightly better is evident in that plot, because we're not taking into account the year difference there. So from our samples that we took for biomass yield, we also measured the chemical composition in the plots, and this table simplifies the data showing the different treatments, so the perennial ryegrass and the 3 multi-species swards, but there was also a lot of variation in the year, the time, the sampling time, the location, and also the calendar day of the samples. But the general trend to pick out from this table is that perennial ryegrass differs slightly in its composition from the multi-species sward mixtures. And these results are from the main 4 agronomy plots, but they are very similar to those that we found on the resilience plots, the medium, wet and dry plots. The other data we recorded was the botanical composition. So these were quadrats that were thrown randomly into the plots and a destructive sample taken, and these were then bagged and then speciated in the lab. So we can see this data here. This is for just 2 of the locations, just as an example of the variation that was happening between the different locations. So we've got cedar on the left and Dutchy on the right, and this set of data is for year 1. And in year 1 we've got a lot of variation, but as you can see from the difference between the colours in the graphs, there were different species being represented at the different locations. So the orange colour here is Timothy, which was doing quite well in our cedar plots, whereas we've got a lot of the darker blue here, which is white clover. So Dutchy did very well on a lot of white clover growth. When we get to the 3rd year of the study, we can really start to see the differences between the locations. So again, the crop control location, as we expect, is largely perennial ryegrass, although there is some unsown species starting to creep in to the cedar plots more so than the Dutchy plots. With smart grass, huge difference between Dutchy and cedar, with Dutchy being largely dominated by white clover that was in the mix, and with cedar having quite a large amount of, we've got still some perennial ryegrass there. We've got Timothy being which was the other grass that was in the mix, doing quite well. We've got some white clover, and we've also got chicory and plantain being represented there for the smart grass. And then we get down to our more diverse species and the differences, again, between the locations, with again white clover being particularly important in the Dutchy plots, and in our drier seasons at the end of the study, cedar is obviously a much drier location than Dutchy. And here it was cocksfoot, this large grey chunk here on the plots that you can see, that was starting to do quite well, and also chicory up here, both quite deep rooting species. So they were really helping support the biomass of the plots when we had the drier conditions. So here we've got our some of our resilience plots. These are the wet location that we monitored, and as we can see here, perennial ryegrass continues to do well throughout the study. So even across to year 3, to the sampling 3, when things were a little bit more challenging, perennial ryegrass is still there as the dominant grass species and one of the dominant species in the mix overall. The issue that we were having with these plots was ingress of weeds into the plots. So we've got a significant amount of unsown species coming into these plots, and this was predominantly Yorkshire fog, which was coming in from the surrounding hedgerow area that was around the plots. Okay, so these are the dry plots. So this is comparing year 1 and year 3, like we did for the wet plots. And you can see that perennial ryegrass does okay in the first year of the project. So the weather conditions were a little bit wetter that summer. So we have still a reasonably, when we get to our diverse mixes, we've got a good number of species represented, and perennial ryegrass is one of them. This situation changes when we look at the dry year. So in the control treatment, we'd expect the blue, the perennial ryegrass, to be there, but as you can see, it was struggling with weeds coming in. So these were things like outside daisy and thistles that were starting to creep into the plot, and docks. And then as we go down into our herbal mixes, the perennial ryegrass almost disappears. So we have interesting smart grass here. This was a treatment which had perennial ryegrass and timothy as its two grass components. So in this case, we've got in the first sampling, the slightly wetter period of time, we've got timothy with some chicory and some plantain there. And then in the later sampling period, we've got almost all timothy in there supporting that plot. The dominant grass for the other two mixtures was cocksfoot, which is a deeper rooting grass, but that wasn't present in the seed mix for smartgrass. So as you can see, the grey colour is cocksfoot, and it's quite dominant in the later sampling, but it's cocksfoot and chicory together that are there for sampling A. And as you can see, we couldn't get a sampling B cut done because the conditions were just too dry and there was not enough growth. So as well as looking at lots of pie charts, what we can do is simplify things a little bit and just look at the total number of species that were present in the mix and see how much species loss we get over time. So if we look at species richness here, and if you remember, we had a 17 species mix, which was the herbal, the blue, a 12 species mixture, which was the green biomix, and we also had a 6 species mixture. So for all of those mixes, even in the first year, there was some loss of species that didn't establish and never came up in the plots. But in terms of species loss, richness loss over time, it wasn't particularly a problem. There was a little bit of a reduction in the total number of species in year 2 for the biomix and the herbal, but when we didn't really see large losses of diversity in the plots. We also looked at the number of rare species, so these are species that are contributing less than 1% of the total dry matter at any point in time. And as you can see, they drop off in year 2 and 3 compared with year 1. And we assume that those species that didn't contribute greatly to the mixture in the first year were then outcompeted in later years. And if we want to think about this for the resilience side of things, the patterns were very, very similar for the wet, dry and medium plots as they are for these plots here. So here's a slightly simplified view of the unsown species, and this is from the agronomy plots with the 4 different locations. So we can see some issues with weeds happening in the first year, which then started to sort of resolve themselves through year 2. And we can see that the increase in weeds that happened in year 3 for those plots. And that's a similar thing as to what happened with the resilience plots. So those dry, medium and wet plots in year 3, the same pattern occurred. And we can see that it's the medium and the dry plots that were contributing to that increase in the control treatments. For the wet treatment, the differences between treatments weren't significant. The Yorkshire fog was just infiltrating across all of the different plots. And finally, we take a quick look at some rising plate meter work that we've got. So for all of the biomass samples that we took, the plots were also measured with a rising plate meter before the cut was taken, and these are some results from that data. And what we can see is the red line there is the control treatment, so the perennial ryegrass treatment, and the other 3 lines are all very close together for the biomix, herbal and smartgrass. So the question is, do we need to have separate equations for multi-species swards compared to control, and do we need to have separate equations for each of the individual treatment types? So if we have a particular seed mix, do we need to think about individual equations for each of those treatments? So not surprisingly, if we start to look at some of these potential factors that might affect the biomass yield, we get significant effects of treatment, location, year, sampling time, and weather variables as well. So they could all be included in a model to improve the fit and reduce the error of those equations. We don't need to go to the level of separating out perennial ryegrass from the different multi-species swards. The model with a simple ryegrass versus all multi-species swards was just as good as the separate treatment model. But there was confounding between different factors, so sampling, location and year, and treatment, and also location and year and treatment. So the best model that we built was one that included the rising plate meter reading, a location, perennial ryegrass versus multi-species sward, percentage dry matter forb, and cumulative seasonal rainfall. But even taking into account all of those factors, there was still error associated with the model, and that average error could account for up to 592 kilos per hectare, even with the best model. So it really does seem like we need to be looking at, where possible, farm specific and possibly time of year specific model equations to help support farmers get the best out of rising plate meters. So a little recap of what we found in this part of the study. The multi-species swards with no nitrogen fertiliser were as productive in year 2 or 3 as perennial ryegrass, and in some cases more so. The chemical composition of perennial ryegrass did differ from the multi-species swards, so we need to be aware of that. Botanical composition varied hugely across sites and across time, with likely effects of rainfall on there as well having an impact. The number of rare species did decline over time, but not the total number of species was not greatly reduced in year 3. And we did have some issues of increasing weed percentage in the perennial ryegrass plots in year 3. And farm-specific equations for rising plate meters are likely to be most accurate. Thank you very much for listening, and please do ask any questions you might have to the panel during the live discussion.